So my name's Roberto, and I'll be talking a little bit about what I did during my PhD. I've since done two postdocs, and I'm starting a company now. So this is old work, but we're still always working on this. We haven't published like the big paper yet, which I'll be talking a little bit about today. So I'm a cellular senescence scientist, and basically this is a very poorly defined phenotype. So mammalian cells, when they are stressed, they tend to arrest. And this arrest is either reversible or irreversible, depending on whether the damage is oncogenic or, if there's DNA damage, to what extent there's DNA damage. And so what's interesting about these arrest phenotypes is that there isn't really a universal marker you can look at in senescence to say this cell is definitely senescent. So we need to look at a panel of biomarkers. And this is already like really annoying because it creates a bit of ambiguity, which I'm going to get into. But anyway, the sort of traditional senescence inducers are telomere attrition. And then if you induce DNA damage via, say, radiation or co-culture with bleomycin or peroxide, this DNA damage is going to induce a senescent-like state. And finally, there's phenotypes that look like senescence where you overexpress an oncogene, and then the cell will enter this irreversible arrest as well. And this happens in various ways depending on the oncogene. For example, MYC will cause the cell to divide very quickly, and this causes replication stress. And this is like a DNA damage response. And this is quite different from what we see when we overexpress like RAS, for example. So when we overexpress RAS, we get this P16 activation and then this sort of heterochromatic rewiring that locks in the cell in this sort of senescent-like state. And this is called senescence, but it's actually quite a different phenotype. And I'll talk a little bit about that as well. But traditionally, these phenotypes have all been called senescence. And this idea that these damage converges on one phenotype has been around for 60-ish years. It's been acknowledged more and more that this is a heterogeneous state, but the way in which we test for senescence phenotypes doesn't really push past that. When we test for senescence, it's like beta-gal staining, some pro-inflammatory markers, and it's a binarized label. The cell is senescent, the cell is not most of the time. So
I'm sorry. I'm sorry to interrupt. Could you hide the Zoom ribbon there because it's blocking the top of the slides.
Yeah, I
There's some way to make it go away. Yeah.
all right, it's kind of hiding now. All right, so there's, as I've alluded, various issues with this definition. So first of all, senescent cells are everywhere. And so this is like kind of a stupid sort of analysis, but I searched for cellular senescence with just a bunch of different mesh diseases and just counted the number of publications we find. And so I only did this for well-studied diseases because there's some very obscure mesh terms which only have like 100 publications in the last 20 years. So we focused on terms that had at least 10,000 publications in the last 20 years, which I defined as like well-studied diseases. And what we find is that 84% of these well-studied diseases have at least one publication linked to cellular senescence in the title or abstract, right? And if we jump to, say, 10 studies, we still get about 50% of diseases. And some of these diseases that don't have very many links, like seizures, which what does a seizure have to do with senescence? Apparently a lot, because if you selectively eliminate senescent cells and rats with induced seizures, they seem to get better. And also like in psoriasis, there's also like only like 2 or 3 studies linking psoriasis to senescence, but we're already selectively eliminating senescent cells in this phenotype in vivo models, and the mice seem to get better, or this seems to help or ameliorate the disease. So these senescent cells seem to be involved in a lot of different processes, and they seem to have somewhat of a causal mechanism, right? But the problem is the lack of a universal marker. So this is from my first study. we built a database of genes that can either induce or inhibit senescence in human cell lines based on genetic manipulations. And we then did some network analysis to identify potentially important hub genes connecting these subnetworks, where if these genes become disrupted, the information flow in the entire network becomes disrupted. So the idea was to try to predict new genes that could inhibit the senescence state. And the idea with a senescence inhibitor is if you inhibit the gene, the cell will enter senescence. So for example, CBX7 was our positive control. This is a potent senescence inhibitor. And when we knock it down, the cell enters senescence, which is assessed by various biomarkers, as I mentioned, because there's no universal marker. So what's really cool is from our predictions, we tested 26 knockdowns, I think, and 13 were like very strong candidates. They were sort of activating a bunch of phenotypes reminiscent of senescence, like with the positive control. But then we get these really sort of weird edge cases where, for example, we get some cells that are actually dividing quicker than the negative control, almost like a cancer. But these cells are staining positive for a bunch of other senescence markers. And you can imagine if you're doing like a histological staining on a piece of liver or something, you might think, wow, there's an increase in senescent cells based on beta-gal staining or based on inflammatory markers. But actually, these cells are maybe even dividing quicker than you would otherwise expect. So this was really annoying to me, right? Because we were trying to classify this in a binary way. Like, does this gene cause senescence or not? And it ends up being not that simple, not that easy to answer this question. So I was sort of ranting about this to a colleague, and he asked me if I'd considered whether senescence could be considered like a combination of multiple phenotypes. And I was like, no, absolutely not. I'd never thought about this. And so we came up with this sort of model where, with the idea that we were measuring different things, different biomarkers that were biomarkers of something. We weren't sure what this something was at this point. And the idea being that there's co-activation of these modules leading to different biomarkers, and we get different combinations depending on context. And this is the heterogeneity of the senescent state. So this is like where the project sort of started. So we wanted to then try to figure out what exactly these sub-phenotypes could be. And so in order to sort of investigate this, I started looking at bulk RNA-seq studies. First, I looked at human fibroblasts. And so these studies were, this is all data from Recount 3. So the data has been sort of like processed together. So it's a bit easier to analyze. And so there was 3 conditions: replicative senescence from telomere attrition, oncogene activation, which causes oncogene-induced senescence, and then this stress-induced premature senescence state from DNA damage, basically. And it's worth highlighting that replicative senescence is also kind of DNA damage. It's sort of programmed within the cell. When the telomere shortens, there's a DNA damage response. So basically, we then found differentially expressed genes between each arrest condition and the proliferating controls. And I'll show a PCA in a second, but this processing involved removing lowly expressed genes and then removing this study batch effect. So we end up with these sort of 4 separate clusters, one for proliferating, one for the 2 quiescent states, and then we get this sort of middle cluster, which is comprised of the replicative and stress-induced samples, and we get this separate oncogene-induced senescence cluster of cells, or samples rather. So we then got these differentially expressed genes. Again, we got differentially expressed genes between each group and the controls, the proliferating cells. And so what we did was we then got these differentially expressed genes and we just overlapped them with different things. So for example, we overlapped genes overexpressed in one arrest phenotype with genes overexpressed in another arrest phenotype. And what we find is more overlaps than you would expect, which is kind of actually expected, right? Like you have genes overexpressed in replicative senescence, and these genes are also overexpressed in oncogene-induced senescence, for example. And we get the same with underexpression. So there's nothing too exciting there. We also have there's a bunch of gene lists that have been published that are purported to be quite useful for identifying senescent cells in single cell and bulk RNA-seq. And the most famous one is Sen-Mayo, which I'll be talking about a little bit. So the nice thing about Sen-Mayo in this case is it perfectly captured the senescent states. So the senescent cells are overexpressing a bunch of genes that are in Sen-Mayo more than we would expect by chance, whereas the quiescent cells are not. So it almost seems like this gene list is really quite ideal for capturing cellular senescence, at least in this context. However, when we start digging a little bit more into the Sen-Mayo data set, there's a lot of issues. So I just wanted to show that this is probably the most popularly used gene list to identify senescent cells. And it's now used as a proxy to identify senescence in aging samples, for example. But the problem with Sen-Mayo is it's got a lot of inflammation genes, actually, and a lot of genes linked to a lot of different processes. So we can do an enrichment of Sen-Mayo using the MSIG database, this hallmark gene list, which are meant to be more specific than doing functional enrichment. And we find enrichment for a bunch of different stress pathways like hypoxia, apoptosis, inflammation, cell cycle, right? And then there's other processes as well that are enriched. And another issue with Sen-Mayo is it's overrepresented within like activated macrophages, for example. So we have classically activated mouse macrophage and alternatively activated macrophages, and Sen-Mayo is overrepresented in both. And so again, this is an example of when a gene list like this can result in false positives. So we'll talk about Sen-Mayo a little bit more in a few slides, but we take these bulk differentially expressed genes and we can also enrich them with the MSIG database. And what we find is nothing particularly surprising. We find enrichment for a bunch of different processes, again, like apoptosis, cell cycle processes. There is a little bit of heterogeneity, such as the hypoxia pathway is not overexpressed in replicative senescence compared to the stress-induced and oncogene-induced senescence. We see some downregulation of lysosomal genes, specifically in the oncogene-induced senescence phenotype, but by and large, there's nothing too exciting here. However, when we consider individual genes, we start to see some more like interesting results. So for example, we can see various inflammation genes that seem to be overexpressed in the stress-induced senescence phenotype and also in this oncogene-induced senescence phenotype, but are actually somewhat downregulated in the replicative samples. And we see various transcription factors which are known to modulate various important processes like P53 and this MITF gene, various FOXO genes. And so this is quite interesting, this idea that these individual senescent states are heterogeneously modulating different stress pathways. And a lot of this seems to be at the epigenetic level. These are chromatin architecture genes. So as I mentioned, there's in oncogene-induced senescence, there's this chromatin rewiring, which involves HMGA1. And so we see that this is overexpressed specifically in oncogene-induced senescence. So it's kind of nice that we can see this from the transcriptome, actually. And again, just the mechanism in which various genes like P16, which is another strong driver of senescence, appears to be overexpressed in replicative and oncogene-induced senescence, but not stress-induced, for example. So again, this is sort of starting to suggest that these are quite different states, maybe. So we looked at metabolic enzymes as well, mainly because we have a metabolism guy on the paper. But it's quite interesting because this prostaglandin pathway is quite, it's been implicated in this heterogeneous secretory profile that we see in senescent cells, because senescent cells will secrete pro-inflammatory cytokines and chemokines, and then other sort of extracellular matrix remodelers. And this process seems to be at least partially mediated by prostaglandins. And so when we sort of map this out, we can see that there's individual enzymes that are differentially expressed in different senescent states. So for example, the stress-induced senescence samples seem to like upregulating these PG synthases that are downregulated in these other senescent states. I don't know if that means anything, but a lot of these are rate limiting enzymes, etc. So maybe just having a few of them be different could be important. But anyway, we just thought we'd sort of highlight that. So we also looked at temporal data. There's this really famous paper in the senescence field where they took keratinocytes, melanocytes, and fibroblasts from humans, and they just irradiated all of them and sequenced the data for 10 and 20 days after radiation. And of course, that proliferating controls. So we did sort of a similar pipeline. We filtered out non-coding genes. We removed lowly expressed genes, and we this time found differentially expressed genes between each cell type and time point and the proliferating controls. And so this is what the three PCAs look like. The fibroblasts at 10 days had some cheeky samples, which were not sort of clustering together. But by and large, the samples seem quite happy to cluster by time point. So, and we captured over 80% of the variance in the gene expression using the top two principal components as well. So we did the same thing. We took genes from fibroblasts that are overexpressed, and we overlapped them with genes from keratinocytes, which are overexpressed, and also with the underexpressed. And we expect to see this pattern. This is actually exactly what we expect. Genes overexpressed in a senescent fibroblast are also overexpressed in a senescent keratinocyte when both cell types are irradiated. So this is very nice. However, with the melanocytes, we actually start to see some very strange expression patterns. We see, for example, that genes that are upregulated across time points in melanocytes, or across the first two time points in melanocytes, are actually downregulated across all three time points in fibroblasts, for example. And so we get this weird opposing sort of transcriptomic profile. I'm not entirely sure why this is. I think part of it is because melanocytes are the cells of the skin that are sort of evolved to deal with sunburn. So maybe when you irradiate them, they're known to start proliferating quicker, actually, at least at first. So maybe this is sort of what we're picking up. But the idea is that these cells are acting very different in response to the same stressor, right? And so we can also look at Mayo again and other gene lists. So for example, genes that inhibit senescence are downregulated in senescence, as you would expect, because these cells are entering senescence, but only in fibroblasts and keratinocytes. We don't actually see these three little red bars in melanocytes. So that's already kind of doing the opposite of what we would expect. We also find that Mayo doesn't actually capture the late time point keratinocytes. So this is kind of like a false negative in this sense. We really went after the Mayo gene list because, again, this is the most popular gene list used, and it does a pretty good job of capturing our cells, but not universally. And again, there are false positives as well. So when we look at processes, it becomes quite interesting. So for example, the melanocytes we have these E2F targets and these G2M checkpoints. So processes involved in cell cycle arrest. So these processes are being downregulated, right? Or they're being downregulated, but not in melanocytes. Again, for whatever reason, they're acting a little different. We see in the keratinocytes that there's differences in the inflammatory response. It's not being upregulated, whereas in the fibroblasts and in the melanocytes it is. And we see the same with this sort of apoptotic signaling. And then we also see differences in, for example, lysosomal gene expression is being upregulated heavily in fibroblasts across all 3 time points, but not in keratinocytes or melanocytes. Same with mTOR signaling, which is just over here. We see that it's being downregulated again in fibroblasts and keratinocytes, but not really in melanocytes until late time points. So in general, we see widespread heterogeneity of individual pathways within these senescent states. And when we consider individual genes, we start to see like very interesting patterns. So for example, lamin B1, this gene should universally be downregulated in cell cycle arrest. And indeed, in our first panel, if I can just go back quickly, we see that lamin B1 is being downregulated across the quiescent and the 3 senescence conditions. But again, when we consider the melanocytes, we kind of see the opposite, that in early senescence or in early stress responses, we see an upregulation, completely unexpected. We can see that the keratinocytes are a lot less pro-inflammatory across time points, which again is, we showed this in the enrichment, but we can see it in the individual genes as well. And P16, once again, is being modulated entirely differently based on the cell type and to an extent a little bit the temporal, the time point in melanocytes as well. We see differences in how various autophagy genes are expressed, various apoptotic genes as well, and of course the transcription factors. So in general, it seems like this heterogeneity is being driven in part by the cell type and how long since the stressor has been perceived by the cell as well. So the reviewers wanted single cell, so that's why we haven't published yet. But this is sort of the latest edition, which is not really in our preprint yet. But we took data from this paper, this diverse and dynamic senescent cell populations, seemed exactly the sort of data we wanted to look at. So they took these human diploid fibroblasts and they were induced into senescence by replicative exhaustion, but also via radiation or etoposide treatment. And so we did a few sort of quality controls based on how many unique genes are being expressed, what percentage of the genes being expressed are mitochondrial, are the cells expressing lamin B1, because you'll remember this is in theory meant to be universally shut down in these cells. And then how are these cells clustering with proliferating controls? So I'll show a few UMAPs, etc. And finally, we filtered for cells that are in G1 or predicted to be in G1 because the majority of senescent states are known to arrest in G1. There's now some G2 examples, but in general, it's all just G1 where arrest seems to happen. So as a sort of sanity check, we also filtered for cells that were predicted to be in G1. So essentially what this means is we only really cared about these cells here for the most part. And the reason for that is there's a large chunk of cells which just are not expressing very many genes. And I don't know why this is. This is, you know, it could be like some of these cells are apoptosed or they're just like super quiescent and not really wanting to move or something. But the point is, if we're not capturing that much information about these cells, it's probably not super useful to keep them in. They'll just look like they're downregulating a bunch of processes, right? Just because they're not expressing anything. So we sort of excluded those. We also made sure that the clusters we were including didn't have high mitochondrial counts. So we see the spike in like mitochondrial counts, but we're throwing away this cluster anyway, so we don't really care about that too much. So from here, we looked at lamin B1 expression, and essentially there's two clusters expressing lamin B1, right? Cluster 0 and cluster 7. And when we look at a breakdown, the majority of cluster 0, 94% of the cells are proliferating, and in cluster 7, roughly 40% of these cells are meant to be proliferating. So I decided to throw out cells in cluster 7 as well, because these cells are replicatively senescent. They're meant to be, but what that really means is you're just waiting until they've divided enough, and it's not always super easy to tell. I think replicative senescence has to be the worst senescent model to study, because you have to wait 6 months or something for the telomere to shorten. And usually you get a fungal infection or something and have to start over. It's like really, really an awful model. But we excluded cluster 7 for this reason, that they seem to maybe be capturing proliferating cells as well. And so what we're left with is primarily cells in G1. And we have a few cells predicted to be in G2M and S phase. So we're just going to remove those as well. And we're just left with this sort of these red cells, which we then renormalize, and we find new clusters with our new subset of cells. And so this is what our sort of final UMAP looks like. And we've got 5 clusters. We've got this replicative senescence cluster, which seems quite different to the others. We've got this quiescent-like cluster, which I'll talk about in a second, why I think this is quiescent-like. And then we've got these 3 clusters, which I named just depending on the breakdown. So for example, the irradiated cluster is comprised 93% of irradiated cells. So that seemed like a pretty good name for it. Whereas the stress-induced premature senescence irradiated cluster is only 74% irradiated cells and 25% at topicide. So that seems to be a good indication that this is more of like a mixed population. Then this green cluster in the middle seemed to be more evenly split. And once again, the replicative senescence cluster was 99% replicative senescent cells. So that was kind of obvious to name it. So we find markers for each cluster compared to all other clusters. And then we overlap these markers with the bulk markers that we found before. And this is a bit different because the bulk differentially expressed genes were generated between arrested cells and proliferating controls, whereas these markers are being generated between different arrested states. So it's a little bit different. But nonetheless, we still see, for example, that in the quiescent-like cluster, there is an overrepresentation of genes that are upregulated in the quiescent bulk as well. So that was sort of our first hint that these cells could be quiescent. And, there's a bit more evidence that I'll show in a second. But the only other really interesting pattern here is that the replicative senescence cluster seems to be sort of upregulating the quiescence markers less than you would expect, whereas these stress-induced clusters are all upregulating these quiescent markers more than we would expect. So for whatever reason, part of the quiescent phenotype looks a bit more like these stress-induced phenotypes in this regard. I'm not sure why that is. It also seems to be that, the stressor is impacting where the cell clusters, right? And so, for example, only 12% of the clusters of the quiescent-like cluster comprises replicatively senescent cells. So for whatever reason, these DNA damage phenotypes seem more happy to quiesce or enter this quiescent-like state. So why do I think this cluster is quiescent? I'll jump back to this slide in a second. Besides the fact that it's overlapping the other quiescence genes quite nicely, it's because we see downregulation of mTOR signaling, and we also see downregulation of DNA repair, which are very indicative. These are very standard processes that happen in quiescence that we don't expect to happen in senescence. In general, mTOR is a driver of senescence, and DNA repair should be increased in senescent cells, whereas in quiescent cells, it tends to be shut off. So this is like another indicator we have that these cells could be quiescent. So just to quickly jump back, I just think it's worth mentioning this irradiated cluster, this purple cluster, is expressing a lot less genes than the other clusters. And I don't know why this is. I don't think it's a technical thing because, again, we only see the irradiated cells really doing this. I think this is like real biology, but I don't know why. Again, the majority of the irradiated cells are not entering this irradiated cluster. The majority of irradiated cells are in the CIPS irradiated cluster. So this is a minority that are entering this state that are expressing less genes. And again, I'm not entirely sure why that is, but it's sort of worth mentioning because when we look at the enrichment, we see that there are various processes that are downregulated, but I don't think this is like real downregulation. I think it's more the fact that these cells are not really expressing very many genes. So it looks like there's downregulation, right? But what's sort of interesting as well is the replicative senescence cluster. These genes are enriching sort of in the opposing direction as some of these stress-induced clusters. So for example, we see this in like G2M checkpoints are being upregulated, and these interferon alpha and gamma responses are being downregulated. But this is sort of the opposite of what we're seeing to these other stress-induced senescent states. So it seems like there could be maybe quite a lot of differences with how these cells are expressing these different stress pathways and other pathways. And there's some differences between the two like stress pathways, the stress clusters, the irradiated cluster and the etoposide cluster as well, particularly with the UV response genes are being upregulated in one but downregulated in the other. And then the UV response up genes are being upregulated in the CIPS irradiated cluster, but not the CIPS etoposide cluster. So it's kind of interesting as well. And we're digging a bit more into this. So the thing is, when this is the same picture, by the way, but when we score the pathway expression in our cells, we can see, for example, with epithelial mesenchymal transitions, even though this is one cluster, we can see that half of the cells are expressing this pathway and the others are not, which to me indicates that perhaps clustering cells like this is maybe not the best way of doing this. So I also would like to do some pseudo time on the UMAP to see if even within one cluster, there are separate sort of fates or decisions being made. Because again, I think these clusters are showing real biology. I mean, they're enriching for separate processes, right? But whether or not these are actually concrete clusters as opposed to gradients or something like this is not entirely super clear to me, right? Like some of this expression seems to be more of like a gradient as opposed to a binary on or off of the pathway, like in DNA repair, for example. But in general, I hope you can appreciate that there is heterogeneity in how these pathways are being modulated dependent on how the cells are clustering, where the cells are clustered. And the clustering seems to at least depend at least partially on how the cells have been stressed. So this is like a very preliminary result, but I tried to do some machine learning, and the idea was to just take genes from one pathway and try to predict cells of each cluster using random forest. And what I find is that there are a bunch of pathways that can predict over 80% of cells into the correct sort of cluster. So for example, just using epithelial mesenchymal transition genes, we can predict 90% of cells into their correct category, which to me indicates that individual pathways are expressed very variably in these senescent substates, if you will. And we can see, for example, the apoptosis pathway is almost capturing 90% of cells, the hypoxia pathway as well, the UV response pathway as well, P53, mTOR signaling are some of the strongest predictors. And these are all stress responses, right? And what's really interesting is we can make a custom panel of genes. I just took 17 genes that are in red. And some of these genes are in multiple pathways, like P21 is modulating a bunch of different processes in the cell, including apoptosis, mTOR signaling, P53 signaling, and some inflammatory signaling. But using just these 17 genes, we actually outperform any single pathway in terms of accuracy, which is like a very small amount of information, right? So I think this is kind of key. The idea that you don't actually need that much information about a pathway. You just need a few representative genes from different pathways. And this is already very powerful for trying to sort of categorize cells into discrete categories. And again, I don't think these are necessarily real biological categories, but it's nice to know that when we divide these cells into these categories, we can still sort of tell them apart based on individual pathways, again, indicating that any individual pathway seems to be very heterogeneously expressed amongst these clusters. So here's sort of the final theory I've landed on. Then the idea is that senescence is just a convenient heuristic. It's a label that captures cells that are stressed. And cells that are stressed are acting in a lot of similar ways. So for example, why do cells that are stressed stop dividing? Well, because if you're stressed and you divide, you now have to expend twice the energy because you have twice the number of stressed out cells, right? You have twice the number of potential mistakes you're going to make when fixing DNA damage, for example. So there's been this strong evolutionary drive from bacteria. You see this in bacteria and plants, yeast, whatever. When a cell is under stress, it will stop dividing. And then on top of this, the cell is going to have to modulate that stress depending on context. What is actually stressing me out? And depending on that, the cell is going to co-activate different processes. And each of these processes has different biomarkers. So for example, beta-gal staining, a very famous marker of senescence, is really a proxy marker for lysosomal activity. And we know this because when we knock down gal1, which is a lysosomal gene, the cell no longer stains blue. And what's interesting about beta-gal staining, by the way, is it's not just a marker of senescence, it's actually a big marker of cancer prognosis as well. So this is actually just a marker of lysosomal activity in general, right? Many of these SAS markers are just markers of inflammation, not all of them. And as I showed, the epithelial mesenchymal transition, these genes appear to be fundamental for sort of the identity of these cells, especially like the replicatively induced senescent cells. When we look at the expression, we can see, EMT, like they really seem to like activating this pathway. So senescence has not evolved in isolation, but a cell that is stressed is going to modulate its environment in some way. And, these EMT genes seem to be part of that way in which they're interacting with the environment. And, senescence is now implicated in wound healing, for example. It's super important in wound healing. If you stop the senescent process in wound healing, then you get chronic wounding. So it really seems to be that these heterogeneous secretory states are important, and we're sort of seeing that a little bit as well with the machine learning, for example. But the main idea is that this model explains a lot of the sort of a lot of the problems we see with the field. So, for example, everyone has been hunting for a universal biomarker of senescence, but I don't think this should actually exist at all, especially if we're just measuring stress, because the stress is going to be heterogeneous depending on what you're actually doing to the cell, what the insult is, what the cell type is. So this explains the lack of a universal biomarker. The heterogeneity is simply activation of different stress response pathways depending on context as well. There's this big sort of paradox in the field of why post-mitotic cells, like neurons, seem to be able to enter senescence when you irradiate them, for example. But if we move away from senescence and start thinking about, well, this cell is just stressed, well, suddenly it makes a lot of sense why the stressed out cell is staining positive for markers of stress, right? And finally, as I showed in the beginning, cellular senescence is linked to virtually all diseases now. But to me, disease is by definition, you're going to have stressed out cells, whether you have Alzheimer's or arthritis, diabetes, or whatever. I think by definition, you're going to have at least local stress on the system. So it's entirely unsurprising to me when you stain with beta-gal the brain of a mouse, your Alzheimer's mouse model, and suddenly it stains blue. You're like, wow, there's senescent cells. Like, no, that's just stressed out cells, really. And that's why we see this phenotype everywhere. So I think this model kind of very nicely explains a bunch of issues the field is having. And I can then just sort of tie this to the literature. So this isn't my paper. This is from Masashi Narita's lab. And this is high-C data. They essentially induce cells into oncogene-induced senescence via overexpressing RAS. And this causes inflammation. And then they also treated cells with TNF-alpha, which also causes inflammation. And they looked at the control of IL-1B. And what they found is that when you overexpress RAS in a cell, I think this was human fibroblasts, there is chromatin rewiring, as I mentioned, these chromatin rewiring that happens when you insert cancerous genes into cells. And this seems to modulate how inflammation is turned on in these cells. And this is entirely different from what is causing acute inflammation from TNF-alpha exposure. So if you're just capturing IL-1B with an antibody, you're going to see it in both contexts. And you're going to say, this is a pro-inflammatory SASP or whatever. But the control, the mechanism in which you're inducing the pro-inflammatory state is completely different, actually. And this could be very important for making drugs that selectively kill senescent cells, because you might be targeting a process that is not targetable in some instances. And, I think this is probably exactly what's happening with many of these senolytics that are not working. And I'll talk a little bit about that in a second. But this is another very interesting paper. This actually won the poster prize for the big senescence conference last year. They found that in mice with cells that are oncogene-induced senesced versus replicatively senesced, they have different surface markers. And one of those surface markers seems to be able to shut down the immune system, actually. So targeting this receptor allowed for the immune system to come and clear the cells. I think they found something really weird, like this only worked in female mice or something super strange like this. And when I talked to the co-author, she was like, I have no idea why that is. They're still sort of digging into it a little bit. But the idea is that if we find markers for what I call stress response modules. Sorry, I call these stress response modules, which are kind of theoretical at the moment. We've not been able to say like these 12 genes are part of this module. But the idea is that these should potentially have universal biomarkers themselves. And if we could find markers of those or ways to target those, we could maybe selectively eliminate cells a bit cleaner. And so why do we want to do that? Because these cells, again, are implicated in a bunch of diseases. So for example, something like 50% of acute myeloid leukemias will recur in humans. And this process seems to be highly dependent on senescent cancer cells, which is in itself kind of a paradox because they're meant to be opposing processes. But again, if we consider senescence to just be co-activation of a bunch of stress pathways, well, suddenly it makes sense that maybe you irradiate a cancer cell, the cancer activates a bunch of pro-inflammatory and anti-apoptotic pathways, but it continues to divide because the cell division is a separate module from the other stress pathways. And suddenly you have a cell that is resistant to treatment and also looks like senescence and also acts like senescence, which a lot of the time is not what you want necessarily because a lot of these pro-inflammatory senescent states seem to actually make cancers worse or aggravate tumors. And what's really interesting, so this paper from 2018, they took a mouse model of Alzheimer's. I think mice are really not a good model for Alzheimer's. But anyway, they took a mouse model of Alzheimer's and then they selectively killed senescent glial cells using a senolytic. And the mice got better. Their memory improved. They seemed healthier. So clearly, these senescent cells, these stressed out cells, are playing perhaps not a causal role, but like I don't know if I would call it root cause of these diseases, but I mean, they're certainly contributing to some extent. And what's really interesting, so my PhD supervisor published this sort of attacking senolytics. And, you know, he's right. Senolytics aren't working, really. They work very well in mice. They work for models of osteoarthritis, diabetes, Alzheimer's, various cancers. But then when we've tried to take them into humans, they don't work anymore. And so if we consider disease like Alzheimer's, like for me, Alzheimer's is a disease of stress. Alzheimer's is now linked to having diabetes, for example, but also viral infections like herpes, bacterial infections in the gums of your teeth, heavy metal exposure, you know, ApoE and tau, which are both of the sort of secreted proteins that get tangled up in Alzheimer's. These are both proteins involved in stress response pathways and not even unique to the brain. I think ApoE is activated in stress across all tissues, actually. So it really seems like cells are becoming stressed in the brain from various different sources. And I find it hard to believe that having an ApoE4 mutation and developing Alzheimer's is the same as having a bacterial infection that somehow leads to Alzheimer's. These are probably different diseases that kind of just look similar, or endotypes as they call them. So I really think that this targeted senolytic approach, or stressolytic, as I wrote in my paper, it's such a stupid word. I think precision senolytics is probably what we're going to rename them. But the idea is you want to selectively try to target these stress pathways. I don't think giving someone a drug that attacks viral induced senescence, if they're diabetic and got Alzheimer's from that, is going to necessarily help them. And I think a lot of the nuance in the field is just based on the fact that there is no nuance. We're just binarizing, saying, wow, there's senescent cells in the brain of this patient with Alzheimer's, without actually characterizing how those cells became stressed and what they're actually doing. So I think the senescence label is now actively harming us, actually. And so I think that's it, actually. So this is sort of what I think the field needs to move towards is trying to actually define these sub-phenotypes of, okay, this cell is senescent. What does that mean? What anti-apoptotic pathways are being upregulated? How is the pro-inflammatory state being modulated? And I think once we figure this out, we can actually start to make actual useful therapies, because right now nothing is really working, right? And as I mentioned, senescence is a hallmark of aging. Senescence is implicated in virtually all chronic diseases and seemingly all diseases as well. So I think really it's time for the field to move away from the word senescence. And I think it's important as well to make the distinction that while cellular senescence, I don't think really means anything anymore, processes like replicative senescence do have a universal biomarker, such as telomere attrition, leading to a programmed DNA damage response and cell cycle arrest. I think this is a definable process. Oncogene-induced senescence to an extent as well, depending on how you define an oncogene. Because again, something like MYC overexpressing in a cell activates replicative stress and DNA damage, and this leads to senescence. And this is quite different from activating RAS in a cell and having these chromatin rewirings. So the point is that those chromatin rewirings seem programmed. The cell seems to have evolved to do this in response to activating an oncogene. So something like oncogene-induced senescence, I'm kind of happy to, like, if you tell me this definitely exists, I would probably agree with you. The other thing is we've never actually seen these heterochromatic rearrangements in vivo. And to get a cell in vitro to do them, you're overexpressing a bunch of a gene, which is not really something that naturally happens very much. So I'm not even convinced oncogene-induced senescence necessarily exists. Masashi Narita's lab did kind of a cool experiment where they took different doses of RAS and injected them into the tail vein of a mouse. And they found that the type of tumor you got, because the mouse ended up with a bunch of liver tumors, it seemed to be dependent on the dose. So to me, that indicates that oncogene-induced senescence is probably also not a binary thing, but also more of a perhaps some sort of toggle. The more RAS you have, the more extreme your reaction to the RAS, and you end up with distinct coactivation of processes, perhaps. But yes, so I think that's it from me. I hope I have convinced you that senescence is not really a real biological phenomena, but actually capturing a bunch of different processes. And that this is interesting because, again, this process definitely is implicated in aging and chronic diseases. So if we redefine the process, then it's worth thinking, why are stressed out cells accumulating with age? What is causing cells with dysregulated stress responses to accumulate with age? I think this is like a fundamental question, actually, and how the cells are becoming stressed as well. I think David Sinclair's lab showed that the epigenetic clock is accelerated, but only in some senescent states, not others, which also creates questions like, okay, is dysregulation of only some stress responses linked to aging? Or are these clocks not entirely capturing what it means for a cell to dysregulate stress pathways? Because clearly they're not capturing the whole story. So I think it's really exciting to move away from senescence and start thinking about this mosaic model. I think it gives us a lot more clarity and also creates a lot more interesting questions. So I think that's everything from me.